Welcome to CureTalks with Gary. Today we'll be discussing novel cancer therapies for immunotherapies with Sellas Life Sciences. Representing Cellus is Dr. Angelos Stergiou, CEO and president of the company, as well as former Chief Medical Officer, Dr. J Sarlis, who will be covering their robust product pipeline. Now, I have had multiple myeloma, and I've had it now for a number of years, and I follow it religiously. And what I have found is that small biotech has been the genesis for every biotech drug for myeloma, and something with dual designations from the FDA, which is like orphan drug and fast track, anything like that, all of those which represent new classes of drug, and each new class of drug provides you with an extra year of life. And when you're talking about when I was first given the diagnosis, I had 33 months to live. That obviously each new year is important. Now, these FDA designations are an offshoot of what we call the Orphan Drug Act of 1983, which was introduced to provide incentives for the development of orphan drugs. These programs have provided over 450 drugs, new drugs, where prior to the Orphan Drug Act, there was just 38 for the over 7,000 orphan diseases. So as you can see, even now, although there's been a heck of a lot of progress, there's still only 450 for 7,000 diseases. This is still such an unmet need, and small biotech fills this need. Cellus has 3 dual designations for their drug GPS, and 2 fast track designations for another drug of theirs called NPS. GPS is a new class of drugs with application across a lot of different cancers. I am pleased to be able to discuss these life-saving developments with the senior level manager of Cellus. Welcome, Dr. Angelos M. Stergiou. We are so pleased to have you here, and we welcome you to provide some talk about the background of the company as well as its history. So welcome.
Well, thank you, Gary, very much. And firstly, I'm glad that despite your initial diagnosis, you're doing extremely well and you're up in good spirits. That's always exciting, and thank you very much for hosting us today. I really hope through your platform here, Gary, to convey an exciting theme of growth and transformation of our company. Our core mission is really to prolong patients' lives. And I'm particularly grateful that despite COVID-19, which is obviously in the epicenter of the news, and rightfully so, we really remain clear in our mission and vision to develop and deliver complex and innovative treatments for patients battling cancer. We're doing really our best to manage, those elements of our business that we believe we can control during this pandemic, that is progressing our assets, generating clinical immunobiological data, and executing really in our clinical development, and progressing with the business development opportunities. So about the company, we're a late-stage immuno-oncology and immunotherapy company. And our focus is really the development of Galinpepimut-S, or GPS, which is an innovative and potentially first-in-class Wilms Tumor 1 or WT1 targeting heteroclitic immunotherapy. What's interesting, Gary, is that the Wilms Tumor 1 antigen in the fetal stage, so before we're born, it's responsible, among others, for kidney formation. Once we're born, it disappears and then it comes back again as a true oncogene. So basically it causes cancer. It's a validated target. The National Cancer Institute actually ranked it as the #1 immunotherapy target to go after via a priori selected criteria. And we in-licensed this technology from Memorial Sloan Kettering Cancer Center here in New York, which is one of the most world-renowned, really, cancer centers, and which is very excited to have such a pivotal partner and collaborator right next to us and have advanced GPS through phase 1 and 2 studies across various indications with compelling translational and clinical data as well as regulatory designations. So we're indeed excited, Gary, that we have commenced the pivotal registration enabling phase 3 acute myeloid leukemia study, which we call the Regal study, in patients after the second complete remission. Additionally, we have a phase 1/2 study that is ongoing with Merck as a collaboration partner. And that's where we combine GPS with pembrolizumab, as well as having a phase 1/2 IST study, which is ongoing with Memorial Sloan Kettering and Bristol Myers, combining GPS with Bristol Myers PD-1, that is nivolumab, in relapse refractory mesothelioma patients. I think we're really excited of where we got. As a matter of fact, it's interesting that this month actually marks the 10th anniversary of Sellas Life Sciences. And interestingly, Gary, Sellas, the wording is comes from the ancient Greek, that means truth and wisdom. And when I founded the company about 10 years ago, my vision was really that through clinical development, through clinical research, we can gain more insight, more wisdom, more truth, and then hopefully get to the finish point, which is to get such an exciting drug approved. My passion and love, if you will, over the last 15 years actually, has been in the immunotherapy and cancer vaccine space, having developed one of the first cancer vaccines in lymphoma. I'm just really excited that with our Regal trial, that is the phase 3 study that is ongoing in acute myeloid leukemia, and the primary endpoint being overall survival. For this study, we expect, Gary, by the end of next year, by the end of 21, our interim analysis, and our previously conducted phase 2 study in this particular indication showed close to a 16-month median overall survival difference in strong favor of GPS. And that was 21 months versus 5.4 months in the control arm with a p-value of 0.02. And we have fast track and orphan designation for GPS and AML by the FDA and by the European Medicines Agency. But what really got us excited, just briefly about GPS and why we entered in this collaboration agreement or the license agreement with Memorial Sloan Kettering, that is the fact that there are about 20 tumor types that express WT1. So indications like acute myeloid leukemia or mesothelioma, inevitably the majority, if not all patients, express WT1. And then we have other indications such as ovarian cancer, glioblastoma, and many other types that anywhere between 30 to 80% of the patients express WT1. So GPS in its core, it's really a mixture of engineered and artificially mutated peptides. And we utilize something called the heteroclitic technology. Back in the 90s, Gary, this heteroclitic concept was used in HIV, AIDS vaccine work. Memorial Sloan Kettering was one of the first, if not the first, institution to utilize that concept and to basically use it in immunotherapy, in cancer treatments. And again, it's by design mutation of the sequence that basically makes the asset, that compound, the drug, more potent and more immunogenic. So basically, it drives more of a stronger immune response that you ultimately need. As I mentioned, we have, it's 4 peptides. We address 25 WT1 optimally selected epitopes that elicit CD4 and CD8 immune responses. So the CD4 and CD8 immune response is very important. Many other immunotherapies that are out there typically target and elicit what's called a CD8 cytotoxic immune response. And that's typically short-lived. And we do both. We do a CD4 and a CD8. So the CD8, if you will, those are the soldiers in the front line. They go after the bad guys, in this case, the cancerous cells. And as I said, they do the work, but unfortunately, it's short-lived. Now, what we have is, we also have a second wave, which are the CD4 helper and memory cells. And basically, the helper cells, they boost the CD8s, the cytotoxic guys, but they also do the work in fighting the disease. And then we have the memory cells, which basically remember what the bad guys look like. So if it comes back, they can fight them off. So basically where we come in, Gary, is that once a patient is in remission, we administer WT1, and now your immune system knows what those cancerous WT1 cells look like, if you will. So if in fact they come back, the CD8 guys and the CD4 immune response, they can go back and fight it off. And basically that prolongs progression-free survival and ultimately overall survival. And what's interesting is we've done a number of studies in leukemia, in multiple myeloma, in ovarian cancer, in mesothelioma. And we have shown really intriguing and exciting both immune biological and clinical data. Again, what we're very excited about is the fact that we're now in acute myeloid leukemia. We're actually in a phase 3 study. I think maybe one more comment, if I may, is that WT1 it resides inside the nucleus, so basically inside the cell. And it's not druggable, if you will, with standard pharmacologic approaches such as small molecules or antibodies. And our mechanism of action is quite unique, the way we work. And I think that's the reason, among others, why we have seen such exciting clinical and immune biological data. So again, the genesis of the company is that we're focusing on GPS, and over the last 10 years, when we started the company, we in-licensed the technology about 6-7 years ago, and we've done a lot of work to progress our lead asset again into phase 3 for leukemia. But we also have a couple other studies ongoing. One is in mesothelioma right now, as I mentioned, with nivolumab, Bristol Myers PD-1, where we're going after mesothelioma patients. And then we have a basket study in collaboration with Merck, where we're initially focusing right now on ovarian cancer. So, we're a small company, a small team. If we had all the resources in the world, we would go after many, many other indications, but we want to stay laser focused. And I think leukemia right now is really our prime focus.
Well, thank you so much. And obviously, I think when I was looking earlier about mesothelioma, their life expectancy is like 8-9 months, and there's really no drugs available. So talk about an unmet need. Now, what I have found is that the FDA has a remarkable history of picking winners in the orphan drug space. Cellus currently has 3 FDA dual designations, orphan drug and fast track. You mentioned those, multiple myeloma, AML, leukemia, as well as the mesothelioma. So that's great. Now, this remarkable accomplishment, this is just a remarkable accomplishment for small biotech. How have you been able to accomplish so much? Because you don't have a huge company. I mean, of the 6 small biotechs that have the 8 drugs for multiple myeloma, you and Cellectar have the smallest market value of any of them. So you guys, you're doing so much more. You 2 companies are doing so much more with so little. So how do you do that?
I appreciate your comment, and I think firstly I have to say that I'm indeed privileged and honored being surrounded by such a strong and smart, intelligent team around me, from legal to clinical to regulatory and so forth. And I think kudos to all of my team. They're really doing such a phenomenal job. I cannot overemphasize the fact that we've had a lot of sleepless nights, and I think our core vision and mission is really to do the best that we can to prolong patients' lives. And I think there's an ethical rationale behind it, obviously, because all of us in drug development, at the end of the day, that's what we care about. It's really to hopefully put our name next to something that's going to be meaningful to patients, and everything else is secondary or tertiary. So again, I really want to thank my entire team, and obviously all of our key opinion leaders and the scientific advisors that we have and the clinical advisors that have helped us really push our asset forward. I think to your point though, Gary, is when we look at GPS, we wanted to make sure that the factors or the differentiating factors it has versus other potential modalities is indeed striking. So I think one is, as I mentioned before, we target the Wilms Tumor 1 or WT1 antigen, and we've done this precisely and deliberately, if you will, because we took fragments from the WT1 whole length protein, and then basically in the lab with it in silico work, in vivo and in vitro, to come up with the best sequence, the sequences that would very likely have a strong clinical effect in patients ultimately, again using the heteroclitic concept as well. I think the second piece is that by using that approach and having utilized 25 carefully selected WT1 epitopes, and we've predicted that those have an optimal MHC class 1 and 2 presentation that ultimately leads to what I said before, the CD4 and CD8 immune response is very important. The tumor microenvironment, Gary, is very important. So as a monotherapy, we go after patients who are in complete remission. I do not believe that drugs like ours are there to debulk disease. That's where you need sort of like the CAR T technology, for example, where if you have a kilo of a tumor, you need a kilo of lymphocytes to basically debulk the disease. We come in as a monotherapy once patients are in remission to then basically prolong progression-free survival and overall survival. But now in combination with, example, a PD-1, we can administer GPS frontline as well as then in the maintenance setting. The other important thing is that we're not HLA restrictive. So anyone walking through the door who tests WT1 positive is eligible to get treated. So be it Caucasian, African American, Hispanic, whatever the ethnicity or background of the HLA may be, those patients are eligible to get treated. And what we've also shown is that we have invoked a multi epitope, if you will, broad cross-reactivity along the full-length WT1 protein that's really suggestive of immunologically mediated cancer cell destruction, which are hallmarks of an effective cancer vaccine. So I think we've sort of put all the right pieces together, but you're absolutely right. I think a company like ours, we've sort of been under the radar, if you will. And I think now with the advancement of the phase 3, we're merging up. Unfortunately, once you sort of fall into the small cap bucket, a lot of folks may not pay as much of attention to you as perhaps they should. And certainly
that leads into the next question, frankly, which is you just did an excellent job of introducing it for me. So thanks. You have one drug in phase 3 trial for AML, and you therefore have, based on the data that I've seen, about a 75% chance of phase 3 approval. Once you're in phase 3, you got a 75% chance of it being finally approved. That's the data. Also, there's another one that says if you're an orphan drug, you have a 73% chance of, if you have a phase 3 drug, to get it to the finish line. So, in my opinion, I look at your SLS, the ticker, And this is not reflected in the potential and opportunity and the value of your company. So, I can't understand it. I am confused, that why that isn't represented. Are there any challenges confronting a small biotech which can be helped to explain this? And like you said, is small biotech just invisible to the market?
Yeah, I think the small biotech arena may be somewhat invisible. I think to Sellas in particular, I think some of the challenges that we had is that we became public by the mere fact of doing a reverse merger. So that had its own, if you will, sort of issues that we had to sort of go through. But I think for a small biotech company to raise capital is a more challenging act versus being a large cap company, and that's just the mere truth to it. But I think what is important is I think that companies like Sellas, like ourselves, we just stick to our true core mission and vision, and we don't deviate from that. I think one thing that we're very clear and have been very clear is that we set our goals and we really execute along those. And I think the other thing is also that you will see from our management team that we're not going to go out and just, for the lack of a better word, just blur out news just for the sake of it. We're very cautious and careful. And I know that our shareholders and investors would love to hear more and more and more and more news. I'm very sympathetic to that. At the same time, I hope that everyone can appreciate being a public company. We don't want to put news out that we don't feel 100% certain that we can indeed at the end of the day back up. So we're very cautious about that. And I think it's also for the benefit of all our shareholders. So I think when we have news, we will announce it. But one thing is certain, that our team is working very diligently, not only to optimize shareholder value, but more importantly, to really, as I said, prolong patients' lives. So I think to your point though, Gary, yes, it is more challenging being in the small cap company because raising capital is more expensive being a small cap versus being a large cap. At the same time, though, I think it's those small companies that, at the end of the day, focus on rare diseases that as you correctly stated, that can potentially, move the needle and provide treatments that otherwise wouldn't be able to be developed. So I think you're on the percentages that you mentioned. Although my mind is very analytical, you know very well that the oncology, hematology space, even the rare diseases, it's more challenging to get drugs approved because by the mere fact that it's a difficult disease state. But I think from the data that we have seen to date, we're very excited, I have to say, and we just really hope for the best, that our ongoing phase 3 trial and all the other indications we're seeking and going after will have positive results. And again, I cannot stress the fact that between our team and our scientific advisory board, we have been there, done that. We know the challenges and we know exactly how to go about that. So unfortunately, there's a discrepancy between the value, if you will, of the asset and the company and what the market cap stipulates right now. And hopefully, over time, we'll be able to sort of reverse that.
Like I said, I'm so confused that they don't at this point, especially when you have a phase 3 asset. I can see it in the first year, second year, third year when you're in phase 1. But when you can see the check, it makes me think that maybe I shouldn't have gone to business school at NYU because it's not. There's no logic or statistics or math that can make me understand why it's not reflected. And I thank you so much for everything that you've provided today. But I think we need to move on now with Nick, if you don't mind. And now that we've talked with Angelo about the background of the company, we want to get into a little bit more detail as to a discussion of the product pipeline for Sellas Life Sciences. Dr. Sarlis, GPS is a drug which has obtained the majority of the FDA dual designations, and its target is the Wilms Tumor 1 antigen on the surface of cancer cells. Is this a new class of drugs, and why has WT1 gotten so much attention from the FDA as an outstanding target?
Well, Gary, first of all, thank you for having me here today. And I will try to follow on from Angelos's excellent introduction of the assets of the pipeline.
That doesn't sound like a Jersey accent. I'm sorry.
No, it's a southern, it's a northwestern Greece accent with a lot of stints in London, Houston.
I was thinking, I know you're from Jersey.
And the upper northeast corridor. So I think, as Angelos mentioned, the vision in Cellus is to build on the promise of cancer vaccines. And we're trying to do that with peptide, novel and sophisticated therapeutics that are targeting major antigens or major targets for immune response to tumors. And one of them is WT1, obviously Wilms Tumor 1. And the other one I'm going to talk in a moment is, I would be remiss not to mention our second asset in the pipeline, which is Nelipepimut-S or NPS, which is a peptide vaccine targeting HER2 in various cancers, particularly breast cancer, for which we also have fast track designation. And we have a couple of trials that are still ongoing and are going to report in the next few months. So, but again, I'm just going to veer back to GPS. And as you mentioned, there has been a lot of attention in this particular asset. So before I do that, I would like to kind of mention a few things about why have cancer vaccines up until recently have been largely unsuccessful in oncology. There are many factors. And I think the cardinal factors is that you have had poor selection of the antigen, right? You have the wrong target. Another factor is that a lot of investigators targeted only one type of HLA molecule, primarily molecules that induce a CD8 immune response, as Angelos mentioned, which is unfortunately evanescent and relatively weak, especially if the antigen is a self-antigen like WT1, meaning that is expressed in early on in normal tissues. The other problem is that a lot of vaccine trials have been using monovalent vaccines, meaning that they're targeting only one or just a few epitopes. And I think a major problem in previous clinical trials with cancer vaccines is that they try to use the vaccine to really kill tumor, right? And they have been using it in the setting of bulky disease, or what we call in hematologic malignancies, blastic disease, especially in myeloid malignancies or fervent myeloma or whatever have you, as a single agent. And we know now through at least a decade or more of clinical immunology work in cancer that you need a lot of lymphocytes to kill established macroscopic disease that is basically visible on scans and whatever have you. And I think the rationale to use monotherapy vaccines has changed and is now focusing on maintaining a remission or maintaining a complete response. And we're going to talk about this in a moment. So I think, WT1 has a number of interesting properties. If you want to attack this particular protein in cancer cells, it is indeed the #1 ranked antigen by a large study that was done by a consortium of the NCI led by Mac Cheever, probably more than 10 years ago now. And it has literally all the properties to be the optimal target for active vaccine immunotherapy in cancer. And one of the things that is very important is that it's not found in an appreciable amount in adult tissues, in adult normal tissues. And this is important because one of the problems that vaccines have had in the past is that you can have on-target off-tumor effect, meaning that you're going to start attacking normal cells in the body, and then you can have toxicities. And this is not the case with GPS. We actually have an excellent toxicity profile, which is basically relevant to immune reactions or local reactions that you see at the site of injection, basically when you get vaccines, for example, against the flu or against the pneumococcal pneumonia. So we haven't seen any systemic reactions that are worrisome or an off-target toxicity, which is actually very, very important. The other thing about WT1 is that unlike many other proteins that are targeted in cancer through non-immune type of avenues, it does not downregulate. One of the huge problems that we have in cancer with targeted therapies, on which I personally have probably spent half of my career, and then the other half was on immunotherapies. With targeted therapies, enzyme inhibitors, kinase inhibitors, and monoclonal antibodies, you have eventually problems with downregulation of the target, and the tumor escapes from the therapeutic attack that you have with this type of molecules. And therefore, with WT1 immune therapies and other peptide immune therapies such as GPS, you actually do not have the problem of downregulation. And I think WT1 is a unique target because it really doesn't downregulate and doesn't get mutated very frequently once the therapy starts. Finally, WT1 is also expressed on cancer stem cells. And perhaps some of your audience have heard about leukemia, cancer stem cells, or even in myeloma, plasmacyte stem cells, and of course, cancer stem cells in general in solid malignancies. And these are the resistant cells that eventually recur and end up killing the patient. And so I think one of the attractive theoretical advantages of GPS is that it can attack not only the proliferating or the progressing cells that are basically continuing to create the bulk of the disease, but it also can attack these cancer stem cells, which is less than 1% of the actual cancer bulk, but are responsible for all the relapses. So I think that's one of the key reasons, and I think also Angelo summarized very nicely, and I'm not going to repeat all this, the fact that it is a highly differentiated type of immunotherapy. We have induction of both CD4 and CD8 responses. You have a heteroclitic technology, which means that you have unique mutated fragments of the WT1 protein that can be much more immunogenic than the native sequences that are expressed on the tumor. And importantly, we are working across a very broad repertoire of HLA backgrounds in human populations. In essence, we cover probably around about 90% of the Caucasian North American HLA, as well as about 80 to 85% of the Asian Pacific Basin HLA. So this is truly a globally applied product. And we also don't need a companion diagnostic because in essence, once you have a tumor that expresses WT1 through standard immunohistochemistry, you can use the drug. And finally, one of the practical considerations that makes the WT1 peptide immunotherapy with GPS very attractive is the fact that this is an off-the-shelf product. And as you know, there is a lot of immunotherapy that is now directed through neoepitopes or neoantigens, as well as the CAR T cells and TRT cells. These technologies are very powerful and they're very promising. However, they are highly individualized and they have a lot of manufacturing challenges and a lot of obstacles as far as distribution and cost of goods. I think one of the practical differentiating factors with GPS that makes it very attractive is that it's an off-the-shelf product with very large applicability.
Well, it's it sounds like it's a brand new target, so it's a new class of drugs, which is great. You now have a phase 3 trial for AML, and it's the closest to approval. Could you provide some of the details behind the trial and the currently available results?
Yes, no, absolutely. I mean, before we go actually to the current phase 3 trial, which I'm going to describe in a moment, I think we need to look a little bit historically how we ended up with AML being as a kind of flagship indication, so to speak, for GPS. So way back then, when David Scheinberg, who's one of the inventors of the vaccine, when he was head of leukemia at Memorial Sloan Kettering. Currently, Dr. Scheinberg is head of developmental therapeutics in the same institution. So when he incepted and conceptualized GPS, he obviously had a great interest himself in myeloid malignancies, both chronic and acute myeloid leukemia. And one of the interesting things about AML is that not only WT1 is expressed almost universally in AML, it also is expressed very densely. It means that most of the proliferating cells, the myeloid blasts in AML, express very densely WT1. And on top of that, the leukemic stem cells are also expressing WT1. So I think it was almost the model hematologic disease to target with GPS. So more than 10 years ago, there was a pilot early study, phase 1, phase 2 study, that showed that there was significant activity in a pilot setting in adult patients with AML who had their first hematologic or morphologic complete remission, called CR1. A phase 2 trial was also performed, which took a number of years to complete and published back in 2018 by Peter Maslach and his team at Sloan Kettering. And quite recently, we actually did kind of an internal meta-analysis, which means kind of merging of the two data sets together, and the results were quite impressive, and I will go over them very briefly. So in the CR1 setting, which is the first complete remission setting, we have treated over the years 31 patients with GPS. These were all patients that had fairly significant disease. They got standard anti-leukemic therapy and ended up having minimal residual disease, which means that they all had molecular evidence of residual disease despite having achieved complete remission under the microscope. And they were fairly standard for AML, spanning with a median age of 64 years and fairly consistent type of mix of cytogenetics as far as risk classification. And what we saw in the pooled analysis, we saw a median overall survival of 4 to 5 years for both trials. The more recent trial was 67 point some months of overall survival. And also, particularly in elderly patients with AML, patients that were 60 years and above, we also saw remarkable overall survival at 34 point some months, which is several fold above what you get with standard treatment. And even in the presence of allotransplant with the elderly patients, we have typically a median overall survival round about 15, 16 months, and we saw 35.3 months, so at least doubling of that survival.
That's
also,
remarkable.
we had, absolutely. Also, we had a lot of evidence, which is, again, very important from a biological standpoint. I mean, it's one thing to see these responses in the clinic, but people may say it's a statistical fluke or it's a small number of patients. But importantly, in all our trials, and this included also these AML trials, we did a lot of immunological surveillance, so to speak, to look at antigen-specific or basically WT1-specific immune responses, and you can do that in peripheral blood, in blood that you can take from the vein of the patients by measuring what Angelos mentioned, the CD4 and CD8 cells. And by doing that, we found that 88% of our AML patients that were vaccinated ended up having immune response measurable by this type of techniques. We also didn't see any effect on any influence on the HLA type, which makes perfect sense because this should work across all HLA types. And importantly, we really didn't see any grade 3 or worse systemic adverse events, which, as I mentioned, is important to have a vaccine that is tolerable and can be given with relative safety. So this kind of completes the package of the CR1 setting. So several years ago, we also did the study at the Moffitt Cancer Center, which is another world-renowned center, with Dr. down in Tampa and with Dr. Pinilla and colleagues. In fact, Dr. Pinilla was working initially at MSK under Dr. Steinberg and then got his professorship at Moffitt, and did a very interesting trial where he took two cohorts of patients that had second complete remission. So these were very advanced stage leukemia patients that had already taken their leukemic therapy. They entered their CR1 or first complete remission and then unfortunately relapsed. And so what happens is they got something called salvage therapy or second line therapy, which, as you know, in myeloma, these multiple line therapies are almost the norm today. But in leukemia, unfortunately, anything after second line is very rare because unfortunately the disease tends to relapse and relapse very vigorously and end up having a galloping type of tempo and kills the patient. So in this very tough population, which is basically second remission population, they did a study where they took 10 patients on GPS and 15 patients that were contemporaneously treated. So these were not historical controls. These were patients that were at the same institution, treated by the same team. And what he found is that the median survival of patients that were treated with GPS was 21 months versus 5.4 months in the control group. And of course, this was highly statistically significant with a median follow-up of 30.8 months. So this is remarkable because, again, these were median age of
myeloma, it's unheard of.
Indeed, was 73. And albeit the particular study, to be fair, was not blinded or randomized, in a post hoc analysis, the patient demographics and the disease characteristics and the type of chemotherapy that these patients had received were highly comparable. So we believe that there was no specific kind of fudge factor there or confounding factor rather that would have produced these results. And again, we found very strong indication of specific anti-WT1 or antigen-specific T cells and immunization of these patients. Of interest, about a third of patients treated with GPS experienced sustained responses and in fact had progression-free intervals that were at least equal to the progression-free intervals that they had in their first remission, which is again unheard of. And of course, this study was published back in 2015, and we were very lucky earlier this year to have the long-term analysis with a follow-up, as I mentioned, of 30.8 months, and we saw this remarkable kind of difference between the two groups. So to cut the long story short, when we put together the phase 3 program, we had a lot of advice, both from our scientific advisory board, but also by external top-line leukemia specialists around the world on how to proceed from a regulatory and clinical perspective to a phase 3 setting. And so with the background of the two CR1 studies and the CR2 study, we decided to go ahead on a pivotal registration-enabling phase 3 study in AML in patients with CR2 or their second complete remission with leukemia with GPS monotherapy. This is called the Regal study. It is designed to randomize one-on-one 116 patients. So it's half of the patients will get GPS and half of the patients will get what is called, quote unquote, best available therapy, which is basically a clinician's choice of the modern therapies that patients typically get in this setting, especially when they're ineligible for or unable to undergo an allogeneic stem cell transplant. So the study is opening in 50 centers across the world. We have about 25 centers in the US, and the other half is in the European Union. We have several certification factors that are important for this particular disease. So it's a very elegantly designed study. We recently got approval from the French authority to run the study in France, and we are expecting any moment now approval from the German Ministry of Health to run the study in Germany. And of course, we have a number of European investigators that are very interested. Suffice it to say, we have had the support of Dr. Hegop Kantarjan, who's the head of leukemia at the MD Anderson Cancer Center, where actually I was associate professor for 4 or 5 years, a few moons back. And Dr. Kantarjan has been responsible for the approval through his clinical trials that he has led both globally and at MD Anderson for more than 8 anti-leukemic and other hematologic malignancy drugs. So we are in good hands as far as running the trial, and his leadership has been truly instrumental. And I will mention that although he's just retired, Dr. Gert Ossen-Koppele from the Hoven network in the Netherlands has been of particular help in designing the trial and helping us looking also at some molecular endpoints and trying to understand the biology of the immune response. And quite frankly, I believe that the trial, the way it's designed and the way it's going to be operationalized and run and eventually hopefully completed will be most hopefully a positive trial based on everything we know about how GPS performs in leukemia and how it should be performing in this particular kind of niche, orphan, and very high unmet medical need population. So we're obviously expecting the results, and we're obviously going to look at them very carefully, but I think we have maximized the opportunity for success here. And again, it's been several years of effort and perhaps several dozen people, both in academia and some of our industry consultants that have been, instrumental in getting us there.
Well, thank you so much for the highlights for small biotech and how it plays such a crucial role in drug development for orphan diseases. What is also amazing to me is the fact that GPS is at least in 20 cancers, is a universal application, which is something that, and I found it with a lot of cancer or of drugs that are for specific cancers. If you look at like Revlimid, for example, it not only plays a role in myeloma, it seems to be playing a role in dozens of other cancers as well. And I think the same thing would apply to you, that once you get it for an orphan disease, all of a sudden it opens up the market and the opportunity in many other diseases. And you already show that you've got that. In addition, it's a new class of drug, and each new class of drug has always been revolutionary, not evolutionary. And what I mean by that is that, you can have, thalidomide, and then incrementally better is Revlimid, and incrementally better is pomalidomide, and on we go, but when you get a proteasome inhibitor and a IMiD, like for myeloma, it creates a far, far better combination.
If I could also interject here, I think you're bringing an excellent point. And just want to kind of perhaps finish this on my end with a little hint on myeloma, which obviously is of high importance to you and perhaps significant.
And all my friends, and
your audience.
It would have been nice for all the friends that are not here today.
Passed. Absolutely. So myeloma, I think, is one of the tumor types or one of the areas of hematologic cancer medicine where we have actually seen this in practice, what you just mentioned, right? So yes, we have the IMiDs, right? And then you have the proteasome inhibitors, right? And then you start having the combinations of the two. And now everybody, God bless, at least in the US and most of the Western world, gets triplets for induction. And soon,
exactly,
maybe getting quadruplets for induction, right? And I think, as you mentioned, and I will corroborate highly on what you just said, the main advances that we have seen the last 3 to 4 years are, what are the advances? They're basically the SLAMF7 CS1 inhibitors. We have seen the CD38s that are very important. We have seen the trail inhibitors that are coming now very, very vigorously in phase 2B studies. And we have seen the BCMA targets, right? And you have the CAR T cells, you have the new monoclonals, we have a number of approvals that are kind of galloping, and I think the FDA has done a tremendous job. And I will also
antibody drug conjugates.
absolutely, and I will also tip my hat off to the European Medical Agency that is very rapidly following this, so that we can make these new medications available to a very broad population. And I will finish that, as you probably know, we had a phase 2 study also with GPS in myeloma, which we found that is very nicely combinable with lenalidomide maintenance. And in this particular trial, we had we not only found excellent immune responses, but we actually found a median PFS in a very high-risk population of 23.6 months. In comparable populations, you have about 14 months PFS. And so we already have a signal in myeloma, which is obviously early, but it's something that we would like to at some point think about, especially now that we have combinations with elotuzumab or daratumumab and other interesting medications that come on the induction phase, and perhaps we can follow them up with combinations of lenalidomide-based maintenance plus GPS plus another drug. And I think we could really potentially move the timing of the relapse much further along, because as you know, once you have a relapse, yes, you can take therapies, but every time that you get second or third or fourth or fifth line therapy, the progression-free interval continues to shorten over time. So I think we have a phenomenal opportunity, especially because, as you also know, the checkpoint inhibitors didn't combine very well with IMiDs, and I think that was, you know, it is what it is. It is what it is, and it has to do probably with some very unique biological characteristics of the immune response in myeloma in specific, which is, as you know, very, very, very complex issue of how people respond to immune therapies and to several antigens in myeloma. But be it as it may, I think with now that we know that checkpoint inhibitors cannot be combined with lenalidomide maintenance or proteasome inhibitor maintenance in early stage disease and upfront disease and newly diagnosed myeloma, perhaps it could be another immunotherapy, and one of these candidate immunotherapies could well be GPS. And so that's the future will show. But I just wanted to ensure that I do not, I'm remiss in not mentioning the fact that we did have some early signal in myeloma as well.
Well, for us, you know, 12 months of additional life is pretty important when you start at 3 years. You know, now we're at close to 6. And what you're saying is that you've got a drug for myeloma anyway that would provide another year, at least, you know, the initial data shows that. And for us, that's pretty important. Another year of life is very important. And it's the people in small biotech that do all the heavy lifting. You know, then you know, when you do the heavy lifting, then somebody comes along and gives you a payoff, and then they take credit for it. You know, it happened with thalidomide, you know, which became Revlimid, which became pomalidomide, which took a little company the size of yours, which was a Celgene, and made it not a $20 million company losing money hand over feet, but to being bought for $80 billion. You know, so you know, little companies can grow up, become big companies. And I think, you know, that's, and when you're late stage like you guys are, you know, it's almost a given. So thank you so much, both of you. I think did an excellent job. Thank you so much to both of you for an excellent CureTalks with Gary.